Good evening, everyone. Welcome to our monthly meeting. Okay, my name is Linda Perry. I am education director. I'm also on the board. We also have a couple of other board members. Joel is a board member, as well as Mark. There is a board member. And is anyone? Oh, Gary.
Me, of course.
Jerry's a board member. Okay, so if you have any questions, ask Jerry.
And Mark. And Mark. And you got Mark in
Or Joel.
the brown shirt.
No.
Yeah.
Okay. First thing I'm going to do is a little update on what's been happening. Some of you may or may not have heard, we're 40 years old. We celebrated our 40th anniversary by throwing a big party. It was over at the American Muscle Car Museum. Anybody get a chance to go? It turned out great. It was very well attended. And now when I go and do presentations and talk about Peter Bondre, I can actually say, I met the man. I mean, I know who I'm talking about now. But he was there, which was good to meet him. And he still looks like it's overdue.
Yeah.
But he was very interesting, told the story of how it all started. And, of course, people were mesmerized. But it turned out very well. And some of the things coming up, I have to cheat because I don't remember everything, is beach cleanup is Saturday. If anybody's interested in participating in that. Nest survey training is Saturday, the 4th. If you've already done it, you don't need to do it again. It's only if you haven't done it and you're a volunteer, a certified been to an orientation volunteer. Then you can do nest surveys. Night walks are next Saturday. Not night walks, but training. Anybody who's a certified volunteer can go and get night walk training if you want. Those are two ends of the spectrum. Nest surveys are very, very early. And night walks are very, very late. So, pick your poison. Or do both. Yeah. That would be cool. All right. I think that covers the things that we had going on. Unless somebody knew of something I didn't, which is entirely possible.
We have
Now
Nature Day, is that next weekend?
the 11th is, oh yeah, Nature Day. Melbourne Beach Nature Day?
Yeah, Melbourne Beach.
Mel Beach Nature Day is the 11th.
And we have our beach cleanups this
I already
Saturday.
said that.
Oh, you did say that.
I got that one.
I notice you don't need to sign up for it. You just show
No, you can. Well, you can if you're a volunteer. You can sign up
Okay.
in Vic. But it's open to everybody. The whole world can do it if they want. Takes a village. It's a dirty beach. So, anybody that wants to do that. All right. Did I get everything?
Sounds good to me.
Anybody have any comments, questions? Okay. So what I'm going to do now is I'm going to introduce our speaker and give her all the time in the world. And this I really need to cheat for because she has creds out the yin yang. Laura is.
Give me the same look my parents gave me.
Laura is an incredibly, she just does so much. But let me give you a bit of her bio. She pursues work that brings together her love of learning, teaching, and exploring. She earned her BS, not literally, degree in geology from Washington and Lee University, her master's in earth sciences from Montana State University, and her PhD in geological sciences from the University of Colorado at Boulder. Did I not say creds? Okay, Dr. Wilson joined the Marine Resources Council. I lost my place. Joined the Marine Resources Council as executive director in June of 24, with a background in paleontology, which is really, I mean, yeah. How many times have I done presentations that said turtles are dinosaurs? Hello. Okay, paleontology, she brings a big picture approach to restoring our ecosystem, looking at how geology, biology, and time are connected. She emphasizes using sound science to guide decisions, building strong partnerships, and fostering lifelong learning so we can create lasting improvements for our environment. May I introduce Dr. Laura Wilson? Yeah.
Please, me.
You're fine, the whole front is fine.
Perfect. Well, thank you so much for having me. Thank you to Joel for finding me and inviting me. I don't get to talk about turtles a whole lot anymore, but I'm very excited to talk about some research that I did right before I moved to Florida and started at Marine Resources Council. So I am the executive director at MRC. So if you aren't familiar, we're an environmental nonprofit dedicated to the Indian River Lagoon. We do a lot of science education and advocacy. But what I'm going to be talking today is actually research that I did in my previous life. So I was a career academic. I was a professor and a museum curator at a university called Fort Hays State University in Western Kansas. So that might seem a little weird to be getting into sea turtle research. But when we talk about Earth's past and we talk about what Earth looked like about 85 million years ago, this is it. So the Cretaceous period, that's the end of the Mesozoic age of dinosaurs. You have the Triassic, the Jurassic, and the Cretaceous. So 145 to 66 million years ago is the Cretaceous. So you can kind of see what the continents are doing, how they relate to the continents today. But we're still in the breakup of Pangea. So the supercontinent that came together about 250 million years ago, when all of the continents made just one big ocean in the Panthalasus, the world ocean, or one big island in the world ocean. And so the continents look a little bit different. Sea level was really high. One of the things you might notice, there's no white, so there's no ice caps. It was a very warm greenhouse world. And then plates, geologic plates were moving all over the place with mountain buildings, which also attributed to why sea level was so high. We can take a little bit look to something more familiar of what North America looks like. So again, this is North America about 85 million years ago. And so while Kansas might not be associated with sea turtles today, I would have had to live on a houseboat about 85 million years ago for where we live in Western Kansas. And so this that covered the central part of North America is called the Western Interior Seaway. It went from the Caribbean up to the Arctic. You'll see the Appalachians were the new mountain range on the block, and the Rockies didn't exist yet. So you had to actually go all the way over to New Mexico to Utah to find the westernmost mountain ranges of North America. And in this interior seaway, this western interior seaway, it was warm because it was a warm climate. It was shallow, so no more than a couple of 100 feet deep at any point. And then the eastern part, so the Kansas part, was actually a lot shallower than the Colorado, Utah part. It had this kind of different cast of characters filling these oceans. And some things look a little familiar and some things look very different. So this was before the evolution of large marine mammals like our pinnipeds and our cetaceans, so our whales and seals and sea lions. So instead of having big marine mammals, we had big marine reptiles. So big mosasaurs, they're related to snakes and monitor lizards. Plesiosaurs, they're like related to nobody, but are kind of lizardy. But then some things that we had these big shelled, pretty much shelled squid or shelled cephalopods called ammonites, great fossils. And then some things that look a little bit more familiar. My favorite description that I've ever heard of the Western Interior Seaway is Hell's Aquarium, because it was just kind of this filled with big animals with big teeth, and you kind of think of it being a free-for-all. This is my favorite piece of art of the Western Interior Seaway by the amazing Brian Eng, and it really depicts, rather than just having these animals kind of cruising by each other, what sort of dynamic ecosystem this would have been with big mosasaurs, the size of a bus or larger, with huge, 12-inch long teeth. And then these big bony fish that we had. So these are these got to be 12 to 15 feet long and were shaped kind of like a tarpon. They're the bulldog fish. They kind of came at you from below. Sharks. So this guy's actually eating a shark. These goofy seabirds that I actually spent most of my career studying, but they're like seabirds, but they still had teeth, but they actually lost the ability to fly. So they're flightless. They're like flightless toothed cormorants that are 6 feet long swimming through the ocean. But then something is a little washed out, but over here is a turtle. Turtles aren't dinosaurs. We actually don't know what turtles are. Turtles are so interesting because they just appear on the fossil record and they're like, we're here, and they're turtles. We don't have a really good transition of them. But we're going to talk about turtles. The turtles that I studied, that this presentation is going to be about, is a genus called Protostega. So Protostega, they lived right before kind of the big dinosaur extinction. So they lived about 80 to 85 million years ago. The big extinction was at 66 million years ago. And they're the second largest turtle known in terms of body size. If we look at our leatherbacks over here on the right, they're about 6 feet or so. So right at about 2 meters. Archelon, which is actually what this guy is the largest one that ever lived. And it's very closely related to Protostega. So Protostega got to about 10 feet long. So think about a leatherback and add another person on top of that. So add another 4 to 4.5 feet on top of that leatherback. And these were what Protostega were. And they lived kind of all over this warm, shallow ocean. They would actually cruise up to the Jersey Shore. So you actually find them up into New Jersey, down into Alabama, which was an ocean at that point. Florida didn't exist. Sorry, Florida. And then on up through the Gulf and up into North America. And so they kind of really made it around. They were well-distributed, well-dispersed species of sea turtles during this time. There's actually even some accounts that maybe they were over in England, but those aren't as well substantiated because it's just bits and pieces of little fossils. There have been a fair amount of studies describing the species, and some of them, when they're first described, were very, very wrong, and they're pretty hilarious to watch the paleontologists fight about. They've been described their anatomy, maybe where they fall in the evolutionary tree of turtles, and really kind of just describing the number of species and what they looked like. But they haven't really gone much further than that. So people haven't really started putting together how these animals lived their lives. So what I term life history. And this is where they meet, when they met, and how they met important milestones in their life. How they grew, how fast they grew, when they reached sexual maturity, so started reproducing, when they reached full body size or skeletal maturity, and all these really important aspects that are hard to get out of an animal that you can't observe living and can't measure as it grows. And so this kind of how they behaved and how they lived their lives hasn't been as well studied as maybe what they looked like. So though I went into this study looking at these Protostega sea turtles, the museum that I had, we've just gotten in this beautiful little, it was still 4 feet long, but little fossil of Protostega. We had a bunch of other ones in the collection as well, and other sea turtle species. So I was really going in this, and I was wondering what their growth looked like throughout ontogeny. So ontogeny is the study of aging, essentially. What happens to an organism between birth or hatching and then old age and death, and how their lives change over that time, how their growth changes. So I was really wondering that as these animals aged, how they grew, what their metabolism would have looked like. These are turtles. We think of them as being slow-growing, cold-blooded animals. The timing of their sexual maturity, so how long it took them before they could start reproducing, and then how long it took them to get to their full body size. And the way that I do this, my animation's off, oops, sorry. The way that I do this is using a term called or a technique called osteohistology. So anybody who knows their Latin or Greek or is a medical doctor, If I know what osteo.
Oh, yeah, we all know that.
Women, osteo. Not cirrhosis though. So anybody know what histo is? Tissue, exactly. So osteohistology is the study of bone tissue. Tissue is a living organ within our bodies that grows with us. It repairs itself over time. And so what I do is I actually take bones and I cut them up. So I don't like to use the word destructive analysis. That scares some people when I ask to borrow their specimens to cut up. I do consumptive analysis, but the information that I get, you can only get by looking at the inside of the bone. You cannot get this information by looking at just the outside. So I put it on some sort of lap wheel, use a table saw for the big specimens, cut it up. I then glue I stopped pointing at the screen. I then glue that sliver that I cut off, glue it to a glass slide, and then grind it down to the point that light can be transmitted through it. And I look at it under a microscope. So this process creates science. And essentially this is a version of what I get. This is actually one of my favorite photos. I did, before I left Kansas, I did a STEAM exhibit. So I showcased a bunch of my research photos as art pieces. And before we did the installation at the museum, I did a little showcase at one of our quarterly art walks. Except I forgot all of my labels for the photographs for what they are. And this was one, and this got the most attention. So if I brought this up, what do you think this is? What does it look like? Well, you're cheating. You look my paper. So the most common ones I got were wood or cork, because it very much kind of has that wood look to it. So this is actually a specimen that I'll talk about. It is a cross section of the femur of a juvenile sea turtle. So you're exactly right. And so this is what it looked like. And so the importance of doing this, even though it does permanently alter a bone, the importance of doing this osteohistological research is because I can look at things like phylogeny, ontogeny, biomechanics of environments. That's a lot of big words. Phylogeny is just evolutionary lineages. Who's related to whom? So a fish grows differently than a lizard, which grows differently than a mammal. And that's reflected in their bone tissue. Ontogeny, again, that's aging. Young animals grow differently than old animals. Biomechanics, that's how we use our bodies, how we use our bones. An animal that swims is going to have a different bone structure from an animal that flies versus an animal that runs. And then environment is probably the least well known, the least well understood and studied of how things like seasonality and climate, as well as behavior, migration, things like that, can affect an animal's growth. And that's really that's been the hardest for us to figure out. So I'm actually really mostly going to be talking about environment as some of these more abstract thoughts for looking at these growth rates. So this is a close-up. This is actually it's still sea turtle, but it's a different species of sea turtle. And I just wanted to take a minute to talk about bone, because probably not a whole lot of people, you learned this in your junior high biology class. Remember this, but bone is, the analogy I like to make is like reinforced concrete, where you have your rebar and your concrete that gives you both flexibility and strength. And bone is the same sort of way. We have two different things that make up bone tissue. And one is, this was just a second. Okay, so one is hydroxyapatite, and that is just the mineral portion. That is the phosphate and calcium that makes up the hard part of our bones. Interwoven between the mineral part of the bone is collagen, which is a protein. And so that's what gives us flexibility and strength. And if you think about baby, how they're kind of bendy, is because they don't have a whole lot of that mineral. They have mostly the protein and cartilage in their bones, and not the hard mineral. So we see these in modern bones, and then either these materials or the kind of echoes of those materials get fossilized as well. The other part that we're going to talk about is all of the open space that we see. And these are vascular canals. These are indeed open spaces of the bone where blood vessels come through And those blood vessels are going to be what's bringing your oxygen and your nutrients to your bone as it grows and or repairs itself. And all of these signals get fossilized, and then both of these signals specifically relate to growth rates. So let's focus on the bone tissue first. We have a couple of different types of bone tissues that we talk about. We can talk about lamellar bone tissue. So this is an alligator, just our alligator mississippiensis that we have here around. And you can see kind of these bright layers to it. And this is showing kind of very organized bone tissue. And it's really showing those protein, those collagen fibers that are just very well organized. Think about they're like little sticks or little logs, and they're all pointing the same way. And then we can go to something like Confucius ornis. So this is a very old, 150 million year old bird that has what we call parallel fibered bones. And you kind of have a row of collagen fibers one way and then a row of collagen fibers other way and back and forth. So it's kind of this intermediate organization. You can kind of see the organizational levels in some of these sketches. And then we have something like Dimetrodon, which is an early mammal type thing. And it has this very chaotic bone where the collagen fibers are going every which way. And if we look at this, I like to think about if we have a time to slow down, take a breath, we have a tendency to maybe like get our self group together and become a little bit more organized. But if the world is chaotic and busy, you're going to be a lot less organized. And so we see this thing with the bone, that your well-organized bone reflects slower growth rates, where your very poorly organized, chaotic bone tissue represents the faster growth rates. So we can get a relative slower versus faster growth. We can do the same thing with our vascular canals. Everything from the top are longitudinal canals, so these are going to be blood vessels that run parallel to the long axis of the bone, to radial canals which radiate out from the middle of the bone. We see these a lot in babies because babies grow really, really fast. So we can also look at the organization of the vascular canals and put a relative, a radial canal growth is going to be faster than a longitudinal canal growth. So we can get relative growth rates faster versus slower. The cool thing about osteohistology is this is the only way that we can actually go in and say how old an animal was when it died. And not like it lived 85 million years ago, but it was 4 years old. And so going back to thinking that this often looks like something that might look like a tree, that most animals actually do lay down growth lines just like trees do. I mean, the Ned-Pollock processes are different. We're not plants, but the same sort of thing. So we can actually go in and count, and this has been well studied in living animals now as well, and say that these growth rings actually represent different years of growth. So this animal was 4 years old when it died. So we can actually then get an absolute age. So maybe it's 10 years old, but was it growing quickly or was it growing slowly to get to that 10 years old? We can put all of these pieces of information together. So let's get back to our turtles. So when I started studying turtles, they actually kind of made a liar out of me because I spent probably the previous 15 years of my career saying that nobody studies osteohistology in living animals except for paleontologists. Because why cut up a dead animal or kill an animal to cut it up when you can just observe it and measure it and put it on a scale or bring out your measuring tape? So that just wasn't a whole lot of data that was necessary for biological studies. But we don't have that sort of data collection advantage in the fossil record. We get one shot of the animal, whatever's preserved in the instant that it died. And so most of our studies looking at modern animals were so that we could interpret them and compare them to our fossil animals and have a point of comparison. And then I stumbled across sea turtles, and there's actually a huge amount of sea turtle data looking at the internal structure of bones, mostly for what we call skeletal chronology. So the exact sort of thing, cutting open bones, counting the growth ring to understand how old that animal was when it died. And that's because sea turtles are protected. I mean, unless you're in the Gulf of Mexico, then they aren't protected anymore, but that's a different issue. But sea turtles, can you send me back?
Uh-oh.
No.
Hey, coffee break's frozen up. So there-
But it's okay. But we have the only way to really understand growth dynamics of sea turtles is to study them after they die. So to actually understand population dynamics, things like when they reach sexual maturity, can start reproducing. We can't go out and kill sea turtles. They spend most of their lives at sea, and it's really just been the last kind of decade or two that we've really figured out consistent radio tagging to get these kind of long life trajectories. And then you have to kind of go out and hope that you catch a tagged turtle to kind of continue their growth trajectory. So much of this, the data about turtle populations, turtle ages in the wild came from studying dead sea turtles, and they do this by aging. So through this kind of wealth of sea turtle data, we've been able to take out that there are two different types of growth patterns in living sea turtles. So one is our hard shell turtles. So everything but a leatherback all goes into kind of the same evolutionary lineage of turtles. So our greens, our loggerheads, our flatbacks, all of those are very closely related. They're in the same family group. And on the other side of that, you might have to advance for me. The other side of that are leatherbacks. So they are an evolutionary distinct lineage. They are not closely related to green sea turtles. How closely or not closely related, we don't know. There's so many questions about sea turtles and their evolutionary history. We don't know. We'll touch on this a little bit at the end. But not only are they not evolutionarily related closely, they grow completely differently. So you have these hard-shell sea turtles, which still have a fair amount of open space that kind of helps with their buoyancy. But then you compare that to a leatherback, which is just this big sponge, essentially, of what their bone is, with all of this open space. And what's really interesting is that if you compare this wealth of data there was for for modern sea turtles, there was zero in the literature for fossil sea turtles. Maybe one or two studies in passing that they were being used in comparison to something else, or a CT scan of a bone, just looking at how much open space was in the bone and how that related to like body weight and the fact that they were swimming versus living on land. So really the study that I got to do was the first one that really looked in depth into how these animals were going using histology. So I wanted to look again at Protostega. And I was specifically looking at specimens from Kansas and the chalks in western Kansas. It's what we had in our collection. It's where most of the turtle specimens came from, but there's actually a fair amount of research, especially with like green sea turtles, that where animals live can affect their growth patterns. So like a Pacific sea turtle population might actually grow differently and at different rates than an Atlantic population, even if they're the same species. So I was trying to control a lot of these uncontrollable factors, and I was only looking at our western Kansas sea turtles. So this was my initial sample size. Paleontology is kind of like the joke of biology, because dinosaur paleontologists have a sample size of 1, and that might be like 1 bone for 1 individual. I had a sample size of 4, which is really good for a paleo study. But yeah, biologists don't like our low sample sizes. But this is what I could get hands on from the collection that I was in charge of. That's one of the advantages of being curator. You can give yourself approval to do whatever research you want. So I didn't have to go through steps to do this destructive consumptive analysis. But I did have the Carnegie Museum as well as the University of Kansas Museum that also gave me permission to sample. So I had 2 small ones and 2 big ones that I was able to cut. So for reference, this big one on the end, it's just this half humerus is about the length of my humerus. So this was a pretty honkin' big sea turtle. Let's start with one of our small ones. So this was kind of one of the best samples that I got. This is actually the same specimen of that femur that looks like a tree. So this is just the humerus, the arm bone versus the leg bone. And so we can look at this and kind of go into some of the details. Ooh, I don't know why that's so blurry. And look at things like there are lots of vascular canals. So all of that open spongy space that you can see on the previous slide. Lots of woven bone tissue and parallel fiber tissue. So these are the faster growing, more chaotic bone tissues, which really shows that the tissue is faster. So it was a very fast growing individual. And then we can go in and actually look at, are there blurry on yours? There's a, oh, no. That's what all of me. Maybe it's my eyes. So the pink triangles are actually pointing to these growth lines. So there are 4 growth lines. So this was growing very rapidly. It was about a 4-foot long turtle at age 4. And that's pretty fast growth for a turtle. And then we can compare that to our largest one in the sample set, this University of Kansas specimen. And again, you see a lot of that really spongy open bone. And we can look at this, again, lots of vascular canals, lots of woven and parallel fibered bone tissue. Again, very fast growing bone. And we can go in again and start counting these lines. This one specifically had 6 of these lines, of these lines of arrested growth, these cyclical and annual growth lines. But that's not the whole story. So one of the things that you see is this is all mushed up in the middle. And this is very common in bones that come out of the Western Kansas chalk. I mean, they had thousands of kilometers of sediment on top of them at one point. And these bones are pretty spongy, so they get crushed. So that middle part is crushed bone. And what we don't know is we don't know if we're missing some of these growth lines, or if maybe because bone is always absorbing as it grows. So as bone grows on the outside of the bone, it actually absorbs from the inside, from the marrow cavity. So we don't know if maybe we lost some of those rings. So I took the previous sample that we looked at and scaled it to the same size. To kind of see what maybe we could have missed. And what this shows us that there's possibly 2 growth rings. Possibly 2 growth rings that were missed due to all of that crushing in the middle. So it's more likely that this was an 8-year-old versus a 6-year-old. But we can look at the full sample set where our 2 smallest were 2 and 4 years old. And then our 2 largest were 8 years old. This humerus, so KU, had 1 partial humerus and 1 full humerus. They gave me the partial one to cut, but they did measure the full one for me. So assuming that the arms were roughly the same size, I actually do know the length of this. So this is showing that the largest specimen in my sample set grew to a very large size, so probably about like a 9-foot sea turtle in 8 years. And the largest humerus, the largest arm bone of this species that's ever been found came from Alabama. And this KU specimen is about 85% the size or the length of that humerus. So if we think about that and actually take this growth trajectory out to that size, then the estimate would be that you would have a full-grown 10-foot sea turtle in 10 years. Which is really, really fast growth. I mean, that's faster than alligators do earlier in their growth cycle. So very fast growing. And interestingly, if you compare the protostega compared to the hard-shelled sea turtles with their one growth pattern and the leatherbacks with their other growth pattern, is that we see that the protostegas were growing much more similarly to the leatherbacks in terms of what their bone looked like. And the cool part of that is leatherbacks are really, really weird. I mean, sea turtles are already weird. And then leatherbacks are kind of like this weird offshoot on top of that. They do a lot of things really differently other than just their shell and their reduced carapace. They have large body size. They're our largest living sea turtles that we have today. They have documented rapid growth. We don't really know how quickly, but we do understand that they actually grow faster and most likely reach reproduction sooner than the hard-shelled sea turtles, even though the hard-shelled sea turtles are significantly smaller. They're exclusively pelagic, which is why we know so little about leatherbacks, is because they spend their entire life out in the open ocean, and they don't have any of that near shore life cycle like we see in the greens when they're young. They do weird things like go to the Arctic. So this blue is showing their range. They go up into the North Atlantic. They really like hanging out those cold water temperatures, which is very weird for cold, quote unquote, cold-blooded ectothermic animals. Usually turtles can't survive in those temperatures. In addition to hanging out in the Arctic, they dive really, really deep. They'll dive 3,000 feet deep. I don't know if the jellyfish taste better down there, but they do. So they go these really deep depths. And then they actually have an elevated body temperature. So this is a study that was done in 1990. And there's a lot of data. But this first line that I'm going to highlight, this is what typical reptile growth is. You look at body weight on the bottom, where lighter ones are on the left and heavier animals are on the right. This is pretty much what you see in the metabolic rate for reptiles. That second line on the bottom is actually sea turtles. So this are greens. So this top line is reptile average. This bottom line is green sea turtles. So green sea turtles are actually a little bit metabolically slower than other reptiles of their size. This area, these are leatherbacks. So leatherbacks holding a resting metabolic rate that actually spans mammals of their size at some points. So they have, even when they're resting, even for a resting metabolic rate, they're elevated. So this is very weird. So this is actually a paper that coined the term gigantothermy. This idea that just the sheer size of the body is actually going to create essentially internal heat, and you're turning into an endothermic animal just by being large size of your muscles moving, of digesting your food. And then this was taken into dinosaurs. There's always a dinosaur catch, right? So this was to show that dinosaurs had gigantothermy because they were so big. So they were essentially functioning as a warm-blooded endothermic animal because of their size. This breaks down for leatherbacks because leatherbacks have really high metabolic and activity rates as hatchlings. They do not stop moving as hatchlings. As soon as they hatch, they're going. You watch a grebe, they'll float for a little while, they'll swim, they'll float. Leatherbacks are all motion. So they do a lot of different things in terms of their biology and their internal biology. And this is one of the things that we have to do in the fossil record, is we have to come up with these modern analogs. And for this, leatherbacks turn out to be a really good modern analog, so we can get some inferences about these life history, these behavior things that are otherwise harder to understand from a mineralized bone. So things like rapid growth to reach large size quickly and rapid growth to reach sexual maturity quickly are things that we can infer about Protostega because their bone growth and their bone growth patterns look so similar to leatherbacks. And we'll talk about these two in a little bit because it's really interesting. Something that sea turtles do is they grow, and then they're like, okay, we're done growing, it's time to have babies. So essentially, when they start reproducing, they stop growing and they don't add body size on top of that. And that'll be something that we come back to. Also, things that are other inferences that you can make on these similarities are the sustained swimming. But I do hypothesize, based on this research, that Protostega had an elevated metabolic rate similar to what we see in our leatherbacks today. So they weren't your typical ectothermic, cold-blooded turtle or reptile. Because I had a collection and a whole bunch of other things in my hand, I didn't just cut up Protostegas, but I actually wanted to look at what else is going on in their evolutionary history. So have y'all seen cladograms, anything that looks like this in other presentations? Okay, so this is showing a family tree, essentially, where these two branches are right next to each other. You can think of those as siblings. We actually call them sister taxa, next to each other. Think of this as their parents. So these are very closely related. because they share parents. Versus this, the Boreacheles, you actually would have to go to back to grandparents to find that most recent common ancestor. So every step back you take, the further apart related they are. The closer they are, and their branches are to each other, the more closely related. So these are siblings, that's a cousin, that's oh my gosh, like first cousin 3 times removed, and they have to go back 3 generations type thing. So closely related, not as closely, even less closely. So the green here, this is the family tree of the Protostegas. So we have Protostega down in here at the bottom. And then these up here, these are, we call it more basal. You can think about more primitive, maybe. It's just they evolved sooner in the lineage. So they're old. They're not ancestors. They're relatives. So they're older relatives, previous generations of relatives. And so I looked at kind of one of these basal, these more primitive, these earlier sea turtles called Desmatocheles. If we look at them on the tree, there are these Desmatocheles padalilii, but I looked at the lowi specifically as the one that I cut. And then you can see that compared to Protostega, which you have at the top of the tree, so these are very derived. These are some of the last family members before the big extinction 66 million years ago. So I wanted to see what these old ancient relatives of these less old ancient turtles look like. But these, these were still large sea turtles. They're still 6.5 feet long sea turtles. So the size of, or maybe even slightly larger than our leatherbacks today. So it's not like they're little tiny turtles or anything. And so I had another humerus, the upper arm bone, in our collection. So I cut this one as well. And you can see that it looks very different from that very spongy bone that we had within our Protostega, as well as the leatherback. So this actually looks a lot more similar to what we see in maybe a green sea turtle or hard-shelled sea turtle, even though we can see that it actually still had this very reduced leatherbacky type shell to it. And this one was growing much slower. You can see how close those arrows are. Those are all of those growth lines. So they're adding less body size each year. So it's growing slower, much more like a sea turtle, or much more like a green sea turtle than a leatherback. And so what I was looking at is you have throughout this lineage this shift from slow growing to fast growing. So as a paleontologist or as a scientist, I wanted to know why. What could possibly be causing within this lineage to go from slower growth to much more rapid growth. So I started looking at what else was going on in the ecosystem at this time. So we're still in the Cretaceous, so 145 million years ago. It ended 66 million years ago. So Desmatocheles, that basal turtle that we just saw that grew like a green, we have them showing up. Oh, I don't know where my arrows went. In the early Cretaceous, that the one that I cut, the Desmatocheles lowi, came, maybe about 90 million years ago. Protostega showed up about 85 million years ago. Mosasaurs shows up between the two. So between the origin of the Desmatocheles lineage and the origin of the Protostega lineage, we have mosasaurs. And if you don't remember mosasaurs, they were made very famous by the first Jurassic World movie as these massive, this is a little exaggerated, eating like a great shark as a minnow, but these huge Hell's Aquarium toothy predators, probably ambush predators, or we'll see in the next slide, maybe even attacking large animals like orcas, where they do a lot of ramming. So we have those showing up about 100 million years ago in the middle or between the evolution of these two lineages. And then right before we have the evolution of Protostega, we have Tylosaurus show up. So Tylosaurus is a specific type of mosasaur that was really kind of the top predator in this Western Interior Seaway leading up to the end of the age of dinosaurs. And so they were very large, very toothy. Remember, they're related to snakes. So they actually have hinged jaws and probably able to gape larger, like a snake does as well. And so these top predators.
They turtles.
In theory, hypothetically, yes. I do not think we have, I'm trying to think, I don't remember reading a paper about, nope, I take it back. The small sea turtle, the 4-year-old sea turtle in this study, it has 40 bite marks in its plastron and its carapace. So probably a mosasaur attack. It's actually, that was almost a complete specimen except for the head and the front right flipper. So probably actually attacked from the front, crushed it, and then like all the gooey stuff just like squirted out the back. But and then sank. So yes, likely predators. However, when you live in Hell's Aquarium and you're surrounded by all these big toothed predators, the best defense against getting eaten is to get big fast. We see this repeatedly throughout evolutionary history. We see this with our largest herbivores are typically larger than their predators. You see this with our whales. Our largest whales are going to be actually our eating off the base of the food chain, are not going to be predators. And a lot of this is because if you get bigger than your prey, and if you get bigger than your predator, it's harder to kill you. So we see this large size and fast growth are a great survival strategy when you have a lot of big predators in your environment. And then because you have body size correlated with reproductive age, so as soon as you get to adult body size, you start reproducing. If you get to adult body size sooner, you start reproducing sooner. So to reach reproductive age sooner and produce more babies is also a great survival strategy. I mean, y'all know better than me. What's the survival rate of sea turtles? 1 in 1,000, 1 in 10,000. I've read both numbers, so it's pretty low. So kind of wrapping this all together, looking at the histology, the protostega, very similar bone tissue patterns, and therefore more likely bone growth and metabolic to our leatherbacks compared to our hard-shelled sea turtle. So probably paralleling that rapid growth to a very large body size. With this also means reaching reproductive age sooner. So you're growing faster, you're reproducing sooner, all great things for survival. And that this may be tied to the evolution of these big apex predators within the ecosystem. Around 100 million years ago, we actually see a big shift in who the predators are. There's a kind of a little extinction event and some turnover in our oceans. And this is really where the mosasaurs come on that have a very different hunting style than their predecessors. And we're probably a lot more efficient swimmers. One of the things, I mean, there's so many questions that I still have about everything. Every science project always ends up with more questions than you have answers. I figure I always tell my students, that's how you know you're doing it right, is you know less at the end of it. But what is actually unclear is one of the things that I mentioned is turtles kind of show up in the fossil record. And we don't have a great transition of what their ancestors looked like and the steps by which they lost their teeth and evolved their shell. And all of those things that come to define what a turtle is. And then they become turtles and they're very successful as turtles. So they don't change a whole lot. They're kind of that definition of evolutionary stasis. It works, so why change it? That being said, there is a lot of genetic diversity, but even with using genetics to determine who's related to whom, we still have a lot of questions. Like, we can genetically sample turtles, but we still don't know how they're related to other reptiles. We used to think that their own little group called anapsids because they don't have holes in their skull. And then we realized that they actually secondarily don't have holes in their skulls. Their ancestors had holes in their skulls. So they're related to lizards. But even now, like, we don't know if they're more closely related to the lizard side of the reptile tree or to the like alligator side of the reptile tree. These are huge debates that paleontologists like to have, depending on what data you're looking at. So we don't know a lot of things about turtle evolution, including where protostega and where leatherbacks fit in, because leatherbacks are not closely related to our hard-shelled sea turtles. So are leatherbacks some way throwback to dinosaur time? Are they more modern and an offshoot? So we can look at a couple of different evolutionary trees. So up here in red, this is our leatherback lineage. Down here in green, these are our protostegids with our protostega. So these two animals grew fast to larger body size and likely had elevated metabolisms. So the question is, how many times did this evolve? So anybody from professor or from doctor know Occam's razor? What Occam's razor is?
Sure.
What is it?
The simplest explanation probably is the right one.
Right. And we do the same thing in paleontology. We just call it parsimony. The least number of hypotheses to get from A to B is probably the correct one. You know, if it looks like a zebra and acts like a zebra, or no, if it looks like a horse and acts like a horse, it's not a zebra type of thing. And so, you know, if we look at this tree, we have two separate evolutions of rapid growth and fast metabolism. Versus there are actually some trees out there that put these things together, where you actually have the leatherbacks as an evolutionary continuation of the protostegids. And all of these things are hypotheses. Every publication that comes out describing a new turtle has a new evolutionary tree of turtles. It's very, very frustrating. But, you know, so some of these ideas, but if you look at this grouping, which is not as common as you see as something like this, but this is just one evolution of fast metabolism. And you don't have to have that kind of convergent or multiple points of evolution happening in the same way. And so there's still a lot of questions. Nothing between these two have been sampled. Even though I borrowed these slides for the leatherbacks from a different researcher, this is probably the most descriptive analysis of leatherback sea turtle histology. So there's just a ton of work that can be done and needs to be done to kind of fill in some of these holes. But, you know, I need a lot of dead sea turtles for my research. So a lot of people to thank for letting me either get into their collections, students who helped me along the way, funding sources as well. So, but thank you all very much. I'd be happy to answer any questions.
I have one.
Yeah.
Did I understand you correctly when you said the leatherback is not related to the archon?
We don't know. So, no, that's too far back. Okay, here. So in this one, no, there are two completely different lineages. We're here are the leatherbacks, and then you have kind of going way, way back. You have the Archelon, which is the next immediate related to Protostega. Over here, you do have them sharing a much more recent common ancestor. So there are other trees that have hard-shell sea turtles and leatherbacks very far apart. So this one actually has, so blue, these are our hard-shell sea turtles, red is the leatherback. So they kind of share a more recent common ancestor. Sometimes you see them on different types of the tree. So like I said, every tree has them differently. So it depends on what study you're looking at. And there isn't great consensus because you have so many extinct groups in the middle and we can't do genetic analysis on them.
Is it fair to say they're closely related to them, but not directly related?
That's more accurate. That smooths over a lot more assumptions. So, and yeah, this one on the right is the only place where you can get close to saying that Protostega is an ancestor. Still over there, you'd have to say that they shared a common ancestor with Archelon or our leatherbacks, but we're very unsure on how closely related
Okay.
they are to each other, or how closely related leatherbacks are to hardshell.
Did you say what you thought the Protostega's diet was?
Probably jellyfish. Very hard to tell because we don't have a huge record of jellyfish. I mean, we have genetics, so jellyfish were around,
Not herbivore.
but yeah, so no, because this was not a whole lot of grass at that time, so there wouldn't have been seagrass.
Oh, okay.
So we don't really see a big evolution of grass until after the big dinosaur extinction.
Huh.
And so those lineages are much younger. So there could have been an ecological equivalent, whether it was an algae or something else, submerged aquatic vegetation, but we don't have a fossil record of what that would have been. So I think, based on the beak, especially, I'm not going to go all the way back, looking at the Archelon, which has that much more kind of elongate snout on it, very similar to like the leatherback shape. So probably more of the predatory versus the grazing. And it's hard because the hook on the beak bill is keratin, which isn't great for getting fossilized. So we're missing some of the non-hard tissue pieces as well. But typically you see them being reconstructed using jellyfish. So, and I mean, that's a lot of protein. So for a big body size, it makes sense. Yeah, diet's very difficult in the fossil record unless you get something with gut contents.
When you said they were kind of leatherback, it was kind of counterintuitive to me because the water's shallow.
I know, right?
So that threw me off. But
It threw me off. I went down this whole rabbit hole at some point doing this research because I guess I just always assumed that leatherbacks had the reduced shell because they were so big and it was a buoyancy thing. And then I went on this deep dive that they actually have a reduced carapace so they don't explode when they dive deep and come back up. It allows their body to compress and then reinflate. And then I was like, well, then what the hell are Prostegas doing? Because, yeah, they live in a seaway that they're not diving at least more than a couple hundred feet. So not like the thousands of feet that leatherbacks. One of the things that I read recently about leatherbacks is that possibly their deep diving is related to the fact that they forage like right off the continental shelf.
Right.
And they actually dive deep to avoid predators while feeding. So it's not necessarily that they're going down to get different food. It's that they're going down to avoid predators. And if those predators weren't there 85 million years ago, then they wouldn't have had that behavior. But then that doesn't address why they have a reduced carapace.
Yeah, sure.
So I don't know why they had a reduced carapace. And if it was the same evolutionary drivers we see in leatherbacks. Because we don't have a great transitional record. And the thing with fossil record is we can only look at fossils and rocks that are preserved and exposed on land. And it's very hard to get deep sea sediments preserved on land. So most of our marine record, fossil record, is shallow marine environments. So it's a bit circular. We don't really know what was going on in the deep oceans and what those ecosystem structures look like. We have a lot of
Where is the active research being done now on turtle?
For fossil turtles, there's still a lot of descriptions going on. A lot of the studies that I've seen come out, there's been quite a bit out of Switzerland. There's a big a big turtle researcher there. And Berkeley used to be a hotspot for turtle, but not sea turtle research. Alabama, there are a couple of researchers because remember the Welcome to the Cretaceous map that Alabama was shallow marine. So they have the same types of sediments that like we had in Western Kansas. So there are some people at like, I guess Auburn has a program down there and University of Alabama has a museum. So there are a couple of people down there. There's a guy in New Jersey who does a lot of sharks, like Cretaceous marine stuff. So there are people that the sea turtles seem to be that like there's nobody that's focused their career on it. It's like kind of a study here, a study there, have access to a specimen. I don't know of a whole lot of big systematic studies happening right now. But there is still a whole lot on modern sea turtles for the skeletal chronology. And I know NOAA does a lot of that research for kind of standardizing the practice, creating the databases and things like that for the modern sea turtles.
On Zoom, we have a question. They said, I always thought turtles didn't have growth rings for aging, but your research seems to prove otherwise. Is that correct?
That is correct. So far, all sea turtles that have been sectioned, and I think 6 out of 7 species, everything but I think the flatback have been sectioned, or maybe the hawksbill, one of the two, have rings. And now that we are able to kind of tag and recapture turtles or capture dead tagged turtles, we can actually compare the ages to make sure that they're accurate. So all of the modern sea turtles have also not only been aged with rings, but also shown the accuracy of using them as annuli, so as annual growth markers.
Cool.
Growth rings were something that was very misunderstood for a long, long time. So again, paleontologists did a lot of this type of research of looking at modern animals. Unfortunately, when we started looking at modern animals, we used really, really bad modern analogs. We use things like humans, which is a horrible animal to use because we modify our bodies and our ecosystem so much. And then we use pigs and cows, which are also horrible animals to use because they're raised in captivity. And again, we kind of change. We didn't use a whole lot of wild animals. So now that we're looking at more species, we're seeing that pretty much everything has a growth ring. Everything except for most modern birds. And they only lack growth rings because almost all living birds reach full adult size within 1 year. So they're going to reach adult size before they would even lay down a ring. But yeah, so now that it's hard to study humans, but now that we study more humans, and more animals, we're really seeing that all animals have growth rings, where originally we thought that growth rings were reptilian and no growth rings were mammalian kind of growth patterns, and so warm-blooded, cold-blooded. It just turned out that we had really, really bad model species for a couple decades. So, but deer, bear, cetaceans, so like our whales, they all have growth rings, mice, other primates, so pretty much cats, dogs, everything has growth rings. And now we even have some birds with growth rings. So like ostriches, king penguins, and they've actually been found in kiwis. But we actually think, so that might be more stress related to birth. If you haven't, this is a complete aside. But if you've never seen an x-ray of a kiwi with an egg before it lays it, I highly recommend you Google it, because that egg is like 2/3 the size of the mama kiwi's body. So it's a lot of energy going into that egg. So maybe one example of the growth ring not being
Ouch.
yeah, I know, right?
Follow up to that question they asked, the aging can only be done after the turtle is deceased.
Correct. Correct. Yeah, it's very hard to get permission to like amputate a leg to cut it up. I mean, I've heard it proposed. My master's advisor proposed it multiple times. He's like, cut off a leg of an alligator, give it a little prosthetic, like. But yeah, so modern sea turtles are all in necropsy, and to this point, yeah, I don't know of anybody like doing a amputated leg as part of a study.
Isn't it difficult to do that on a bird since birds' bones are out?
You just have to be careful when cutting them, but just because they can shatter if you don't like decalcify them correctly. But cutting them isn't hard, it's interpreting. Because remember, so birds' bones are hollow, has to do with the flight. But again, as we add more bone to the outside, so our bones grow on the outside surface. And as they grow on the outside surface, they absorb old bone on the marrow side, the marrow surface. So birds, because their bones are so thin, they're constantly absorbing all of that previous growth record. So we're getting just a snapshot of right before they die because everything else has been reabsorbed, versus the turtles, which don't reabsorb as much from the inside of their bone. We get a much longer growth record and can go back multiple years. But yeah, birds are hard because of that for sure. That's why I did flightless birds, and they have thicker bones because they don't have to worry about flying. They get more of a record.
And dolphins, whales have growth rings in their teeth. Have they been found to have them in their bones based on this work?
So animals, okay. Secondarily aquatic animals. So animals that originally evolved on land, well, they went from like water to land and then back to water, essentially through their evolutionary history. So things like dolphins, whales, cetaceans, pinnipeds, they came from, like whales came from hippo type animals. That's their closest relative, the hippo. So something on land that then transitioned to secondarily live in water. They all handle that adaptation to kind of the neutral buoyancy differently. And they can handle it differently at different times of their lives, like whether they're a baby or whether there's an adult. The two ways to handle it is either you get really, really dense bones. So really, really thick bones that can help you see. If you've ever seen a manatee bone, it is a dense bone. Versus some of them get really spongy bones, kind of like we see in the leatherbacks and what we see in a lot of adult cetaceans, so especially as they get bigger. So yes, they have growth rings, but they differ very differently at different parts of their lives or different parts of the evolutionary tree. So they can be difficult to find. And there has been even less work on cetacean histology than there has been on sea turtle. Yeah, cetaceans are big, man. They're big. And there are a lot of cool things about them. But yeah, so teeth. And so you're also getting into mammals. So a lot of mammal studies use teeth for stable isotope geochemistry, where you can get to things like diet and climate and things like that, and aging. And as mammals, like whales, you get one set of teeth. You have your deciduous, your baby teeth, and then you shed them, and then you have your adult teeth for the rest of your life. So you have that continuous record in your life. As reptiles, so things like dinosaurs, mosasaurs, plesiosaurs, they're reptiles, so they continuously shed their teeth. So we can't really say that like that's their lifetime. That might be one season. Maybe they spent half that time in the Arctic and half that time in Kansas, and swam between. So it's a lot harder to use teeth for some of those studies because they're constantly being shed. And then you get to turtles and they don't have teeth. Another things that you see people use are otoliths, especially in, or I guess otoliths in the ear. So especially in fish, it's kind of a bone in the ear that adds layers to it. So that's something that's used. And then I can't remember which animals. Some of them use like plates in the eyes as well. So some animals, especially that live underwater, actually have bony plates around sclerotic rings. So actually bony plates around their eyes so that they can transition pressure. So those also lay down rings, fish scales. So there are a lot of different ways that you can do this on the animals.
Lots of thank yous and fascinatings and very interesting online. And a happy Passover and a happy Easter from someone online.
Thank you.
Lots of thank yous. I'm just making sure I didn't miss any questions. And more fascinating.
Do you have a question?
I just had one more.
Yeah.
Has this kind of analysis been performed on other marine creatures from that time? Like, was everything growing fast at this time?
Not necessarily, because I mean, we had sea turtles. I didn't talk about this. I actually cut another one. So up here, we have even less of an idea of where something like Toxocheles was evolutionarily, but I cut Toxocheles, which lived at the same time as these guys, and they had very like green sea turtles, slow growth.
Yeah.
So even within our turtle diversity of contemporary species, we had them living very different lives. So I had a bunch of students that were doing mosasaur, so the big marine reptiles, mosasaur histology, and trying to piece together series. And a lot of that was looking at endothermy. So, I mean, things like mosasaurs, like a lot of our secondarily aquatic animals were probably warm-blooded. So would have had more of that kind of mammalian growth rate.
Oh, interesting. Okay.
Yeah. But things are weird. I mean, it's so cool. Like alligators, like we now have a lot of evidence that alligators are secondarily ectothermic, that they actually evolved from an endothermic warm-blooded ancestor. Because going back to the Triassic, the crocodilians, they were like bipedal, so running on two legs. And then they went quadrupedal. And then when they did that and became like semi-aquatic, they became ectothermic.
Wow. Weird.
Yeah, so like there's some really cool stuff that's coming out right now.
Are there great movies that we can watch and see?
Well, to see this.
Because I've
There is a new, there's a new one that just came out. I can't remember what streaming platform it's on. But
But just that clip was so cool.
Yeah.
Oh, the dinosaurs?
Yeah.
Dinosaurs one?
Yeah.
Yes, I just started watching.
And then a year ago, David Attenborough actually narrated a documentary style dinosaur one.
Yeah.
I've been watching.
And then like Darren Naish, who's a well-known paleontologist in England, I think he was the consultant on that one. So I think that one's fun to kind of watch them in a scientifically accurate documentary style, like as if you're watching them in nature.
And they have like
Yeah.
accurate depictions.
Yes,
It's really cool.
the
I
animation.
think I saw that a couple years ago.
Yeah, and that one's on Disney. Or no, that one might be on Apple Plus.
Oh, I love that.
Yeah.
Well, thank you. I love that clip. I was like, oh.
Yeah, that's stress. That's the first stress.
Don't you love it? They're mostly scientifically accurate.
Yeah.
Isn't that was that?
No. In terms of the size depictions, because the shark hanging is a great white. So if you assume that's like a 16-foot shark, you're now at, like an 80-foot mosasaur. We do have them. So like the biggest Tylosaurus, the one we had on exhibit at the Sternberg, we had a 30-foot on exhibit. And I think Tylosaurus maybe got up to like 40 to 45 feet. And then Mosasaurus, which that was supposed to be, maybe 50. I don't even know if we got up to 60. So that one was like a good probably 20 feet, like school bus longer than anything. So that one, yeah, that was a little exaggerated. But the idea is that they're all retro-engineered. They've all been modified in some way, so you don't have to keep your historical accuracy if they're all genetically modified. So that was the workaround that they worked that one around. They finally explained that one in the fourth movie.
Okay.
So, but my master's advisor was the technical advisor for the first three movies.
Oh,
So
Wow.
got a little of the behind the scenes there.
How many are there now? How many?
I just
Like
watched
five?
the last one.
Six?
Oh my God.
Oh, seven. I think Johansson one. I think that's the seventh one.
One came out in 25.
Yeah, I think they rebooted it again
Mm.
with
I gotta
Scarlett
see some.
Johansson.
But they're scary.
You just can't do that first one.
Netflix.
Huh?
You just can't be the first one. The original Jurassic Park, there's a whole bunch of us. I was 12 when that came out.
Yeah.
And we're the Jurassic Park generation. That was very influential for us. Kind of showing people that paleontology is a career and kind of putting things into the limelight. So there's been a huge flood of students kind of following that really started hitting grad schools in the early 2000s. Yeah. But it's like it got people talking. It got dinosaur scientists. Like these are real careers. These are jobs that you can do. And then people started thinking about them that way and pursuing them. So now we just have more students than jobs, but it's pretty typical.
And here I'm from the Flintstones and the Jetsons.
There you go.
And we have a new vocabulary word. Gigantothermic.
Gigantothermic. Yeah. which was, yep, so it's used for dinosaurs. It was originally coined to describe the metabolism of leatherbacks.
And this will show up on the next.
Because, I mean, that was a huge debate for decades for dinosaurs, is were they endothermic or ectothermic? So cold-blooded, warm-blooded, or warm-blooded, cold-blooded. And really kind of histology and now stable isotope geochemistry are the only way we've been able to answer those questions. And some of it goes back to the misinformation of mammals don't have growth rings, reptiles do. Dinosaurs have growth rings, so they grew like a reptile. Now we know that to be false. And so this idea of when did endothermy evolve within the dinosaur lineages, because birds are dinosaurs. And the same way that a cat is a mammal. And so we're all dinosaurs warm-blooded, which we actually think they were now. And we actually think all dinosaurs had feathers at some point. Like pretty much every major lineage of dinosaur has some sort of feather or filament covering their bodies. And one of the reasons for that, like why mammals have hair, is for insulation, for when you're endothermic.
Okay.
And so like birds didn't evolve feathers to fly. That has nothing to do with flight. They could fly because they had feathers, because they had them for insulation and probably for mating displays. It's all about sex mating. All evolution. That's all like this.
Let's see if that better.
They had I mean, they're mammals, so they have fur, they have hair.
I know, that's what I meant.
Yeah. And then pterosaurs, so flying reptiles, they're not birds, they're not dinosaurs, they have pycnofibers. And so they have little fibrous growths on their wings. And so now their questions are those like evolutionary the same as feathers.
As soon as I get that time machine.
There you go. I know this is gonna take all the fun out of it though. Like to actually be able to thing. We can't just speculate them.
I'll just dump you back in the past and hey, have at it.
That's okay though. It'll be so tasty.
I know.
How fast are you?
I know, right? There's nobody next to me, just to be faster than him.
That's true.
Well, excellent.
Thank you.
Thank you.